we are very fortunate, Pierre, to have you. I mean, you were actually labeled by St. One of all the people as the Gandalf of vaccines.
Yeah, you guys get Gandalf without the beard today.
And we are actually very fortunate to have you because of why? Because of, if I show you this slide here, this is really the reason why you're called Gandalf. You have a tremendous background. More than 30 years, I think, Pierre?
Yeah, 35 years in the industry after 3 years of residency in a hospital doing clinical pharmacology. So that's more years than I would ever admit.
Okay. We have a cross-section of people today, some people with a medical background, some people with a pharmaceutical, some people with a business background. So if you'd like to...
yeah, we can get started. So we'll skip the video, right?
Yes, we'll skip the video. We have a short video, but I'll share that later with the participants.
Okay, lovely. Well, thank you all of you for joining. I'm really honored to have such a distinguished panel. I don't think I know everybody on the call. I know some of you. It's actually nice to see you at least connected or in tiny post-temp size videos, but still it's very nice. Let's get on. Just one last thing, maybe one housekeeping detail before I forget. So many of you were connected with me, and I know I had some feedback recently that they could not find me on LinkedIn anymore. Well, there's good reason for this. I have been bounced for being arguably too harsh on an anti-vaxxer who was actually giving me hell on something that he had no command of, and for some reason, maybe I should have hold my pen a bit more delicately, because I've been sanctioned by the LinkedIn police. But this is now resolved, and now I have a new profile up and running. So those of you, but I lost all my contacts. So those of you who are connected with me, feel free to reconnect with me on my brand new profile. Now let's move on, which is, the disclaimer, you can forget about the disclaimer. It's, since I'm working as a director in listed companies, the disclaimer is almost a must-have in any presentation. So basically, this is the agenda that we're going to try to cover about, a few insights from 2 years of pandemic management, because we're already, 2 years into this situation, whether there were some winners or losers, at least seen from the vaccine perspective. I understand there are some of you have been asking questions, and this is why it is fitting there, whether we should go for a booster shot, and if so, when and which one. Then I'll evolve into a little bit of elaboration on what are the other applications for those novel vaccine technologies, and what do we need to anticipate in terms of preparing for the next pandemic. Maybe, Salman, can you put this into full screen?
Yes, please. That is, again, my mistake. Is that better?
Yeah, that's a lot better.
Okay.
Can I have the next slide, please?
Yep.
So your next slide, please. So basically, can I get it next? Yes, thank you. So remember, it was in March 2020 that the WHO declared the coronavirus epidemic, which the first cases were diagnosed in 2019, which was called COVID-19. And what you can see is the world has faced several waves of infection, and this is the CDC data, so that's US epidemiology. And you can see clearly a disease coming in waves, but not necessarily aligned with the season, because when you look at the main wave in the center, it is clearly centered on the winter months. But then when you look at the next wave, it was actually starting peaking in the late summer. So there is a seasonality which is not as well established for the coronavirus as it is for influenza. Next slide, please. And I couldn't resist starting with a little joke here. It is, because we've picked the Greek alphabet. So this has been probably raising a number of PR concerns for some brands, ranging from Corona Beer to Delta Airlines to Omicron nanotechnology, and hopefully we won't get to the Swiss watches, and we'll find a solution to that problem before we get there. Next slide, please. But it is still evolving, and what I wanted more seriously to point to is that, and this is the most simplified version of the genealogy of the coronavirus, the so-called SARS-CoV-2. And as you can see, some of the lineages have been generating various offsprings. Some of them have been completely disappearing from the epidemiology. Some others are still there and still evolving. So it is still a moving target. But it is important to keep in mind that why do we pay so much attention to this is because the variants have differences in terms of how contagious they are, how fast and they circulate, how severe is the disease that they trigger, and possibly how much of an escape to the antibodies they could generate, meaning would they render the existing vaccines less useful? Next slide, please. And so all those policy questions are really on the table. Once you have the phase 3 trial of a vaccine, the phase 3 trial results, you would think, okay, we're pretty good and pretty safe. And the reality is not so much, because of a number of other questions that come downstream, revolving around manufacturing, maintaining the quality, maintaining the output. It's been a real issue to not only produce enough doses, but also to pack them. We've been short of vials, we've been short of all sorts of consumables for manufacturing. And then in order to get to the poorer countries, you had to obtain the WHO certification, not necessarily the PQ, but the emergency use listing. And then you had all the logistics constraints. And all those questions, then culminate in all the questions about how durable is the immunity? What's the schedule for boosters? Do we know enough about the core rates of protection, meaning what is the magnitude of the immune response that you need to have in order to be clinically protected? How effective are the vaccines? How sustainable is the protection, etc.? And all this on the backdrop of an ever-changing target. So essentially, this situation has been keeping all the manufacturers on their toes, because it's not that because once you have finalized your phase 3 that the work is done and you can move on to only manufacture and commercialize. Next slide, please. Now, also, there's still a question that we're grappling with. It is still a relatively recent publication. What you can see is we have a world where we have a very unequal access to vaccination. What you have on the left-hand side is a heat map where the color corresponds to the percentage of coverage, and as you can see, there's a huge imbalance, especially in the low-income countries have been very poorly supplied. And what you have on the right-hand side is a number of doses per 100 people, and a scale goes to 200, and you can see high-income countries are very well covered all the way to boosters, whereas low-income countries, not everyone has received doses as of today. Next slide, please. So also on top of this, it's quite important to keep in mind that the pandemic has had a massive economic and social impact. I think the economic impact is visible there. This isn't just an example. This is the change in employment in the population in the G20 countries, with a stratified by age on the left, gender in the center, and education on the right. And as you can see, massive impact on employment, largely on women, and across all types of educational background. I could have picked also a graph on the impact on schooling for children, but I think what we need to realize is beyond the medical impact, beyond taking care of patients, this pandemic will have lasting social and societal impacts, and we will probably see the ripple effects 10, 20 years down the road. So we need to keep this in mind when we design policies for the management of future pandemics. Next slide, please. Now, addressing quickly the question of winners and losers, when you look at, of course, which vaccine were administered on the left-hand side, and you look at, European Union, okay? So arguably Switzerland is not part of the EU, but many of you are in the EU, or maybe not the UK, sorry, not anymore. I see Andre smiling. As you can see in Europe, it's largely a Pfizer monopoly, okay? There's a bit of Moderna, and then the rest is almost immaterial. Look at the worldwide situation on the right-hand side. It's predominantly AstraZeneca. Next in line is Pfizer, and you do find then in the top 10, you do find the inactivated vaccines from China. So winners and losers, it depends on which geography you're looking at. And if you're the Minister of Health of Germany or the Minister of Health of Rwanda, or of Ecuador, the situation is very different. And we need to also keep this in mind because it has a consequence, and I'll come to this in a minute. Next slide, please. So when you look at those novel technologies, what I tried to do is I picked a graph, I stole a graph from, can't sure what it is. I think it is, Airfinity. So you see, Airfinity is a consultancy that works with WHO and that provides, data about the sale of, various products. And what I've been representing with this, ugly little shapes, the red pentagon, the yellow hourglass, and the blue diamond are the nature of the technologies. And as you can see, you've got technologies that were not in use in vaccines or not in large use in vaccines, respectively in the case of RNA and viral vectors before the advent of this pandemic, which are now in the top 10. Well, you got 3 viral vectors and 2 RNAs. The rest being more traditional technologies, inactivated vaccines or recombinant proteins. And I think it is important because when we talk also about winners and losers, this pandemic has accelerated the access to 2 novel technologies that hold fantastic promises for the future. One of them being RNA, the other one being the viral vectors. Next slide, please. So now that we're in a situation where I hope all of you have been vaccinated. If you've not been vaccinated, you probably had COVID, maybe non-symptomatic. So in any event, keep in mind that your immune system has already faced some imprinting from COVID, whether from the vaccine or from the disease. Okay, even if you are not symptomatic. So the question becomes, at a time where we're entering into the winter, at least in the North Hemisphere, where the influenza epidemic is giving early signs that it will be pretty severe, which is not good news, and where the hospitals are already overwhelmed with patients, especially at the 2 extreme ages of life, infected with RSV, shall I get a booster, and which booster shall I get? And it starts by, of course, trusting the vaccines. So I think it is important to make sure that the population understands the relative risks and benefits of each of the technologies to make an informed decision, unless the decision is made by the policymakers in view of the public. Next slide.
Yep.
Sorry, I'm just realizing that this slide could have been placed into the previous section. But anyway, I think the point here is, for the first time in vaccine history, you have 4 different technologies to address the same pathogen. If you take, for instance, influenza, you're using hemagglutinin cultivated on eggs. You got a very small fraction of hemagglutinin generated by, cell culture. So if you take Haemophilus B is bacterial fermentation, and then you conjugate the polysaccharide with a carrier protein. Stop. You just have different types of carrier proteins, but that's it. Here, it is the first time that for the same pathogen, you got 4 and soon 5 different technologies to address it. The whole virus vaccine, the RNA, the protein, and the viral vector. And each of these technologies come with their own strength and weaknesses. So if you take only the 2 recent ones, what is the benefit of RNA? Very good antibody response. What is the downside of RNA? Very poor T-cell response, and therefore a poorly sustainable immune response. Therefore, the need to re-immunize regularly. Viral vector, on the contrary, gives you a kind of okay antibody response, but a stellar T-cell response, therefore a very good memory, and therefore a more sustainable immune response. You would argue that you would need to combine the 2, which is exactly what I did for me, by the way, starting my prime with a viral vector and then getting RNA in the end. But I did my own little cuisine. It was not allowed in Switzerland, so I had to travel to get the first shot of viral vector out of Switzerland. Close the parenthesis. I think right now it is extremely difficult for the authorities to agree on what is the best vaccine and best technology we should use for boosters. And I think it is still a legitimate question that everybody should be asking himself or herself. If you're asking my opinion, what I will do for me, I will not get another booster of RNA. I will seek a recombinant protein because I don't need another viral vector because I had one already. Next slide, please. Now, the vaccination policy so far has been quite, efficient to, and this is Swiss data. So those of you living in Switzerland will be interested there. What you see is what is over time the progression of the coverage overall, and then by age, and you can see that the older age group was covered prior to the younger age group. No difference in terms of gender, some difference in terms of place of residence. And overall, a little bit of a correlation between the income and the level of coverage in the latter part of the pandemic, where essentially people with lower income were having a tendency of being less properly covered. But what is quite interesting is that now higher education and higher income contribute to higher vaccine booster adoption. And this is only Swiss data, but it is the case across a number of countries. And there's an accumulation of papers showing that they're essentially sociology papers. They're actually demonstrating that the people with lower education on average tend to be more vulnerable to vaccine hesitancy because they're reacting more, they're clicking more with anti-vaccine propaganda, which is quite worrying from the perspective of the public health authorities, which of course try to maintain a decent level of coverage in order to, avoid again having the healthcare system completely overwhelmed by an influx of patients. Next slide, please. Now, when you look at what are the main reasons for vaccine hesitancy, again, this is Swiss data. So, I'm not picking on any other country than mine, so there's nobody should be offended there. And when you look at and you get a big of a difference by gender between male and female, what is actually quite, interesting is that men question more the efficacy of the vaccines, whereas women tend to be a bit more afraid of side effects. But otherwise, you do have a substantial fraction of the people who prefer so-called natural or traditional remedies, and If you accept those, I think all the other questions are very legitimate. I've had the COVID infection. Do I need to get a booster? If nobody explains to me why I should get a booster, why would I get it? Right? If I have questions about the efficacy of the vaccine, why would I get an injection if I don't trust this medication to be efficient? And of course, if I'm afraid of side effects, I will definitely see the side effects. And I have no reason to, if I think I can resist the disease, or if I think it's a mild disease, I have no reason to take action. So again, there's a lot of education that needs to be deployed in order to, overcome those doubts that contribute to diminish the level of trust that the lay public is having in vaccines. Can I get the next slide, please? Now, we're talking about mix and match and novel technologies, and I'm at the forefront of one of them, so I'll be happy to elaborate on one of them. I don't want to give the impression I'm advertising. Mix and match is something which was recognized by the SAGE, the Strategic Advisory Group of Experts of the World Health Organization, as early as February 2020. What is mix and match? It is about using a different technology, a different vaccine for the priming and for the boosting. Because vaccine science is telling us that if you do so, you get a more amplified response and a more sustainable response. Which, by the way, is exactly what I did, and a number of colleagues of mine who've been in the vaccine world for long enough. They've all been doing their little cuisine even before it was authorized by the government. Okay. Now it is increasingly the case, but it is also the case out of necessity, because in some cases, the countries were not able to get the vaccines they wanted, so they had to implement mix and match. Now, you do have now the emergence of an alternative route of administration, such as the so-called mucosal vaccines, and you see two examples there. One is the intranasal, where you see a little syringe-like device to actually spray the vaccine to the nostrils, and it gets into the cavum, and then elicits an immune response, starting by the IgA, so which is called mucosal immunity. And what you have on the bottom is the CanSino's nebulized vaccine, which is actually nebulized. It's an aerosol. It's prepared extemporaneously in a cup, and you see the subject essentially inhaling through the mouth on the picture on the right hand side. Now, you may have seen that AstraZeneca has published recently results saying that their intranasal was quite disappointing. They were actually eliciting an immune response with their intranasal, which was below their injectable. Whereas in the case of CanSino, going through arguably a more complex procedure, which is the nebulization, in droplets that are below 2 microns in size, what you get them in the lungs, and you get actually a very good immune response, both locally and also circulating immunity, which is higher than the injectable. Why would this difference be? If I'm quoting the lead researcher from Oxford, who was the head of the clinical program for AstraZeneca, he was saying that maybe when you spray in the cavum, if it goes, if the vaccine is carried in the mucus and swallowed, it gets destroyed in the stomach before it can elicit a proper immune response. But anyway, the point being that nowadays there's a lot of research on mucosal vaccines. Why? Because those will be actually more acceptable, especially by the people afraid of the needle, and they're actually very well tolerated. The side effect profile is extremely favorable. Next slide, please. So basically what we are doing now is the combination of a search for the suitable formulation. So will we end into a situation looking like influenza? Maybe, maybe not. Why? Because it has been demonstrated that the original vaccine against the Wuhan strain is extremely protective against the latest version of the Omicron variant. And I'm not talking about BA5, BA6, because those are already outdated. So I'm talking about the progeny of BA1 and BA2. So really now what we're looking at is because of the speed at which this virus is evolving, maybe we'll evolve towards more what do you call multivalent vaccines, where you would have maybe several proteins or several target antigens, not all of them linked to the receptor binding domain, in order to elicit a pan-coronavirus type of response. Those are still under development. Next slide, please. And the reason why it is so important is, and this is only, of course, a graphical representation, but I think it was quite elegant. Every individual in the population is having a different immune memory, because it has a different immune history, if you will, depending on whether you had the infection in the first wave or whether you did not have the infection and you got vaccinated, and then you may have been exposed to the virus in the second wave. So and it is also represented in the next slide. It gives you what is called an immune imprinting, and the immune imprinting is different depending on the individuals. And what you want is to have a sufficient cellular response, so that your T cells represent a library of epitopes, if you will. They will recognize all the cousins of the family. So if they see anything even remotely looking like a pathogen, they would react by stimulating the B cells and then having antibodies produced. Next slide, please. Well, this is exactly the same thing. So let me move on to the next slide in the interest of time. So again, as I said, the trend is towards bivalent boosters, at least. But at the same time, those bivalent boosters, as I said, are a little bit outdated at the time when they're commercialized. Oops. And then...
Sorry, just a minute.
Tony, you're not on mute.
Tony, I just asked you to mute yourself, please. I think it's muted now.
It's muted now. And so what you have is, you will have, of course, a harder choice for the individuals. If you get the choice, which one should you get? And again, it is very reassuring that if the time comes for you to get your booster, and the only thing you can lay your hands on is a vaccine corresponding to the old formulation, well, it's been demonstrated that this older formulation is still very good at protecting against the currently circulating Omicron variants. And you don't need to go to extremes in order to get a bivalent booster. But if you do get a bivalent booster, of course, it's very good. Next slide. So then again, as I was saying, the real key question now, which is the question that all the authorities are grappling with, and there's not a good alignment across countries, precisely for the reason I was explaining before, because each country is having a different history in terms of procuring the various vaccines and using it in different proportion in its population. How do you integrate the COVID-19 booster in your overall vaccination scheme? Number one. And number two is, which booster should I recommend? So right now across Europe, the prevailing opinion is we should use RNA because those are validated as boosters. And you need to know that there are a number of technologies, knocking at the door of the regulators. And therefore, the situation is likely becoming a bit more fragmented going forward. Next slide. So very quickly, because in the interest of time, what could be the application of those novel technologies? If you move to the next slide, because RNA and viral vectors are new, but they are actually essentially emerging as promising platforms to address other diseases. Now, before COVID, RNA was used essentially in the field of oncology immunotherapy, trying to elicit an immune response against tumor-specific antigens. Viral vectors were already used in vaccines against Ebola. They've been tried unsuccessfully in vaccines against HIV. Next slide, please. But again, I think there's a flurry of candidates. So I'm not going to get into the details of the slides. You'll get the slides anyway. But I just wanted to show you a graphical representation stolen from McKinsey about the number of targets that are currently investigated. Against both infectious diseases targets, but also cancers and even rare diseases. So we're probably at the dawn of course, there will be multiple failures. We're probably at the dawn of an era where there will be, novel solutions for diseases that were prior, that were not addressed in a satisfactory manner. Next slide, please. Now, when you look at the VC capital funding going to biotech companies from 2019 to 2021, okay? I just picked this, those are the numbers, and I've just been using the same, ugly little things, the viral vectors and the little icons, okay? And you can see where you find those technologies in the cell therapy segment, in the next generation gene therapies, in immuno-oncology, and in infectious diseases. So as you can see, for the investors, the pandemic has also served as a gigantic de-risking exercise, which is translating into a reorientation of some flows of financing towards fields in which those new technologies are being used. I find it quite interesting. Next slide, please. Yeah, and then of course, I got a few slides on pandemic management, which is one of my pet subjects, and I'm involved into working groups up to my eyeballs. So I'll be happy to share some of the dark reality of what it is. Next slide, please. But essentially, we know that we will have another pandemic. What you have on the right-hand side is a chart showing over the years the number of spillovers from animals into humans. Arguably, you would see that there's, compared to the 1900s, you see probably an acceleration of the problem, and we know that we will have to work on laboratory surveillance, on regulatory coordination, on manufacturing platforms, on mock-up dossiers to accelerate the filing. So all those activities were, by the way, already on the table in the aftermath of the 2009 H1N1 pandemic. But since that pandemic ended up being relatively mild, a number of those activities were shelved and gathered dust, and they were essentially recycled this time around. So hopefully this time around we'll be a bit better in getting ready for the next pandemic. Next slide, please. Yeah, and again, I wanted to just illustrate that now everybody is, focused on coronavirus, and we probably have something like 7 billion people on the planet who are experts in coronavirus. Mark my words, influenza remains a much bigger threat than coronavirus. Why? Because coronavirus has a genome which, mutates because it's the viral replication is a messy process. It's not a very reliable process. But the genome is actually more homogeneous, whereas the genome of the influenza virus is made of tiny segments, and that thing recombines like crazy. The H1N1, influenza that you see on the right-hand side, which gave, in 18 was plain vanilla bird flu, but the one that was circulating in 1957, the so-called Asian flu, was a hybrid between bird flu and human flu. Okay, and the H3N2 is a combination of H2N2 and H3. Influenza can combine and then recombine into a number of nasty things. And I'm telling you, this one is being monitored. The good news is the surveillance network, which was already in place around the world to monitor the emergence of new strains of influenza, is currently being leveraged to monitor the evolution of the coronavirus variants. Next slide, please. So there's, and I'm not going to spend too much time on this one. What you have on the left-hand side is a screen capture of the grand challenge organized by the Gates Foundation earlier. I would say, no, this one was actually last month at the end of October, about pandemic preparation was very high on the agenda, and you have multiple subjects revolving around prevention, detection, and response. And I'm not going to delve into all of them, but of course, prevention is all about identifying and surveillance. The detection was the surveillance. Prevention is about creating vaccine solutions, and response is about being able to deploy these solutions as fast as possible. It will result into a fragmentation of the manufacturing footprint of vaccines, with moving away from a small number of large factories to a large number of smaller factories spread around the world and close to where the population is. Next slide, please. And that actually takes me to the discussion. I just wanted to finish on schedule, so I will have time for questions. And you'll have, when you receive the slide deck, you'll have my directions on the last slide, which is the one next to this.
This one? Yeah.
Yeah, this one. And on that note, I'm sorry it was maybe a bit rushed, but hopefully you enjoyed the ride, and I'm happy to take any questions.
Pierre, thank you so much. Thank you so much for an excellent presentation. And there are a lot of questions that we will be posing to you. The first one is coming from Tiziana. Tiziana, do you want to unmute yourself and ask your question, please?
Yeah, can you hear me?
Yep. Yes, Tiziana, yes, we can hear you.
Thank you.
We'll both hear you and see you, by the way.
Perfect. So I have many questions, but just to allow everybody to chime in, I have one. How much do you think that the governments are prepared for everything that you've said around the preparation, so the prevention, the reaction, and everything that will be the next waves? Because we are facing still an economic crisis, a war, and everything. So, how much do you think that the investment is in the government's agendas?
I'm not involved intimately in the preparation of the budget of each of the governments, but I would say that typically governments are less prepared than they would like the people to believe, or that they tell their people. Case in point is the situation when you have the minute you have a wave. And you saturate the emergency or urgent care or intensive care is a clear sign that the level of preparation is not at the level that is required. Now, I think some lessons have been learned. I think it is now quite evident that things such as the basic principles of hygiene, washing your hands, not touching your face, wearing a mask, all this works reasonably well. Lockdowns are probably not so useful, except maybe more selective lockdowns of the frail, of the seniors, or etc. Anyway, we now know that and by the way, we knew it before, that the schools are the best petri dishes. Typically, the kids pass on the bug to each other, and then it moves into the households from the children. Tiziana, I am sure you know why Italy was the most hit country at the beginning of the pandemic.
Tell us, please.
Actually not.
Well, because when you look at the demographics, Italy is the European country where you have the highest proportion of households where 3 generations are under the same roof.
Wow.
The grandparents take care of the kids, walk them to school, do the groceries while the couple is working. And that was lethal.
Confirm. Yeah.
Interesting. Pierre, thank you so much. There are a number of hands which are up, but I'd also like to, if I may, perhaps bring in Daniel Paris, who is the head of medicine, as some of you might know, at the Swiss TPH. Daniel has been traveling the world. He was in Tanzania. I think he was in Bangkok last week. Daniel, can you hear us? Daniel, can you hear us? He was muted, but one of the things that I wanted. There you are, Daniel. Can you hear us?
Yes, I can. Sorry, it's a bit. Yes,
no problem. No problem. Thank you so much. And one of the things that I wanted to bring up, as Pierre mentioned quite eloquently, the inequality in vaccines, and this is something which I know is also quite close to your heart. It seems to be the case that probably there will not be an end to this inequality, Daniel. I don't know what your comments are, if you have any questions, Pierre, on that.
Well, I mean, it's a major issue. And I think the initial attempts were overly enthusiastic and optimistic, and unfortunately did not turn into reality as much as we expected. I think the COVAX initiative learned us a lot of lessons. And clearly, much more emphasis has to go into low and middle income countries. I think also development of vaccine, not necessarily the whole vaccine, but even vaccine parts could and should be transferred into countries with lower economies with our support. And I think this would greatly improve access to vaccines in these countries. I think the map that Pierre showed, it is so impressive. It hasn't changed much since the beginning of the pandemic, where Africa is just a big red continent amidst a sea of, So it's really a major problem. I mean, we at Swiss TPH are very concerned about this. There are quite a lot of activities relating to this, but unfortunately, these mills turn slowly, and there is no really fast solution for this.
Can I just briefly echo the comments made by Daniel? I think the COVAX scheme was a wonderful idea in the first place. It's just that it just happened that the countries which had the monies just were ready to buy the vaccines, and the manufacturers actually played this supply to the countries against the supply to COVAX. Also because in order to be eligible to COVAX purchase, you would have to be WHO qualified, meaning obtain the emergency use listing, which entails a full inspection of your facility by WHO and a complete review of your medical dossier and an opinion of the SAGE Strategic Advisory Group of Experts, and all the rubber stamping, and all this moving at the WHO speed, okay, which is that of an elephant with rheumatoid arthritis, and so in the end, what you have is this asymmetry between the need to react fast and a pretty heavy administrative process. That's point number one. I think the second point I'd like to echo, though, that Danielle is, which is very dear to my heart because I'm involved in some of those projects, especially through my board role at Univercells. I think the real solution lies in localizing production in some places, but we will not be able to localize it everywhere. Why? Because you don't only need the equipment, okay? You need the machines, etc., but you need also the people. Meaning you need to have trained personnel, and not just trained personnel for production, but also for quality control and quality assurance. Now, you can't just find, trained professionals just everywhere simply because the politicians are deciding that you should put this here or there. And so in the end, some of those projects, having vaccine hubs here or there will survive, and those will die. Why? Because you need to keep them what is called warm and ready in between pandemics. Now, what do you actually keep these people busy with in between pandemics when the technology that you're localizing is having only one application, RNA, or a couple of applications, viral vectors? The good news with viral vectors is the manufacturing technology and the quality control is pretty much the same as producing viral vaccines. So, cell growth, cell-based growth, etc. I mean, all this is relatively similar. So you do have some, I would say, manufacturing similarities, and you can keep the knowledge, the know-how of your team current. For RNA, right now, there's only one application. So I think one of the merits also of discovering new applications of the RNA technology will actually create the foundation for the sustainability of localizing this production in emerging countries. And again, not every emerging country will be able to do that in a sustainable manner. But if only, I don't know, even 10% of the current projects are surviving, that's already a quantum progress compared to the previous situation dating from prior to pandemic.
Thank you so much. That's excellent. Daniel, any further follow-up question to that?
Just a follow-up comment. I 100% agree with Pierre, and we've had many a debate around many of these issues. But I just want to add on to the GMP aspects of production in LMIs in low and middle income countries. You have to also realize that, the vaccine supply chain is extremely important in the whole, if you look at the big picture. For example, the RNA vaccines, with their storage temperatures, pose substantial challenges to actually roll out, for a vaccine rollout initiative. Whereas if you have a vaccine, which might be a nanoparticle-based vaccine, which is storable at 4 degrees or at room temperature, or a protein vaccine, so that there are, it's more than a 3-dimensional problem which we're tackling with. And I think this was a real, a global wake-up call. And between the pandemics, as Pierre rightly says, now we should be hyperactive in being ready for the next one. So have a network of production facilities which are not, too dense. I mean, better to have fewer, but with GMP settings, and really promote these. And obviously, local capacity building, staff training is a key element to making this a successful endeavor. Yeah,
fully agree.
Thank you, Daniel. By the way, for all the people on the call, we will be organizing something quite ambitious with Daniel and the Swiss TPH, hopefully sometime early in the year, next year, on how to improve access to medicines in less developed countries. And that's going to be probably a half-a-day event that we will be putting together. Sanfana has been waiting patiently on her hospital bed. Sanfana, do you want to ask the question, or are you able to ask the question, Sanfana? Or should I repeat that? Oh, good.
Hello,
I can hear you.
Hello, Salman. Can you hear me? I'm afraid there are bells ringing in the background, which might be overpowering my voice, but can you hear me?
Yes, we can hear you very well.
Wonderful. Moreover, can Gandalf hear me? Because here, I was imagining in my mind, Gandalf in the caves of Mordor with the fire demon approaching, which is COVID, and your weapon was, the vaccines, and you're very much the gatekeeper, telling us really interesting perspectives about the whole wonderful battle we've fought. I think it's worthy of another talk. Just seeing how we've put off such a huge disaster that was coming towards us. However, one of the major worries that people have is the side effects, that whether we've had the proper time to evaluate side effects in individual people, because these are very powerful drugs. And so I was just wondering, very powerful chemicals, and I heard a lot about the spike protein playing a role in inflammatory processes, leading to increased inflammatory responses in different individuals and cardiac events. And I'm just wondering what you have to say about that, Pierre, in terms of side effects faced by individuals and what we should expect, whether we should have more warning, whether we should be more careful, whether dosing has to be changed. and things about children and frail individuals, whether one size fits all, whether we really need to tailor things moving forward.
Thank you, Sanwana. Lots of questions.
Yeah, all those are excellent questions. I'm going to try to be to summarize the response. I mean, first of all, it's always the same goal, which is try to have the right balance between efficacy and safety for any medication, and is the case for vaccine. Now, at the beginning of the pandemic, when you don't know, what is the what will the lethality rate be, the so-called case fatality ratio, CFR, you go for efficacy at the expense of safety, which, by the way, is the route that Moderna took. When you look at the dosing of the Moderna vaccine, it's no wonder that the booster is only half a dose, because it's almost a veterinary dose. 100 micrograms of RNA, it's a huge amount compared to the 30 micrograms with the Pfizer vaccine, okay? And those of you who have received a Moderna shot, like me, I've certainly some recollection of being for 3 days in bed, shivering and having a flu-like syndrome, which was actually quite characteristic of the side effect profile of that vaccine. Now, as recently as this morning, I was reading a publication showing that it is now demonstrated that there is a higher incidence of myocarditis in juveniles with the RNA vaccines. Okay, so this is now, when you get to population levels, and billions of doses used, and in countries where pharmacovigilance works well, you start to discover a few things. Will there be longer term effects? It's hard to say. In the case of most vaccines, the side effects materialize relatively soon after the injection, no more than 6 months. So now, in principle, the risk of long-term things is probably pretty remote, although it cannot be eliminated completely. Now, I think the question you were raising is about the spike protein and the contribution to an inflammatory process. Yes, I've been reading a few papers on that subject, but at the same time, it's very difficult to distinguish the role of that spike protein being produced in the inflammatory process, because anyway, by definition, you want to trigger inflammation as the beginning of the immune cascade. If there's no inflammation, there's no immune response, to put it very simply. And as a consequence, what you have is it's okay to have inflammation and to have fever, because what you want that. I mean, this is the sign that essentially the immune cascade is being triggered. Now, in the specific case of the RNA, as you know, the lipid nanoparticle formulation, which is needed, otherwise RNA would be degraded by the enzymes and would not get into the cells. So the lipid nanoparticles are there just to make sure that it gets into the cells. They play the same role as an adjuvant. Most recent adjuvants are actually lipid emulsions, and lipids are not very well tolerated. They're pro-inflammatory. So, in other words, the formulation of RNA is in itself pro-inflammatory, in addition to contributing to what it is there to deliver, which is facilitating the penetration of RNA in the cells, and as a consequence, that amplifies, the inflammatory response. So I realize I'm not fully answering your question, Santwana, but again, I think the reality is we had vaccines that were actually doing their job in terms of protecting the people. Is there room for improvement in the formulation or improve the safety or the safety efficacy ratio? Yes, there is, clearly. And there's a lot of work currently ongoing to reformulate, especially the RNA, into non-lipid nanoparticles or into exosomes, extracellular vesicles, or other essentially solutions that would do the job of protecting the RNA, do the job of facilitating the capture by the cells without having the side effects and the pro-inflammatory side effects.
Okay. Thank you so much.
Thank you very much. Yeah.
Yep. We have a number of hands which are up. Rachel, you first, and then Christian, and then Shashka. But before you get to them, the questions from Rachel and Christian and Shashka, a couple of quick fire ones. Can pregnant women safely get a booster vaccination or not? This is a question from Fabian. And secondly, if if you are already taken your third or fourth booster in my case, the fourth shot in my case, should you then also get a flu vaccine? Because you said before that flu is the main suspect. So first of all, pregnant women, and then flu together with the COVID vaccine.
Yeah. So pregnant women is the perennial question. I mean, first of all, it is many vaccines are given to pregnant women, not to protect the woman, but to elicit an immune response in the mother, so that the mother antibodies would find their way into the baby, and the baby would be protected in the first months of life by the antibodies from the mother. Okay, so it is not uncommon to immunize or to give vaccines to pregnant women. Now, in the specific case, I must admit I've not been reading a lot dedicated publications on that specific subject. If I was to do it, I would actually recommend using more, like time-tested technologies such as the recombinant proteins or the non-replicating viral vectors. I'm God knows I'm a staunch pro-vaccine activist, but I would not, venture in using RNA in a pregnant woman.
Okay. Thank you so much. Fabian, I hope that answers your question. And then what about combining COVID with flu?
So combining flu, there's already data out there. You could actually go and get the flu in one arm and the COVID on the other. Apparently, it is well tolerated. There's no interaction. For anybody in this audience or in your family who would be senior, frail, diabetic, etc., by all means, don't bypass the flu jab.
Yep.
Don't bypass the flu jab. Again, the first signals that we're getting from the US, especially first signals are pretty worrying, with cases of influenza diagnosed so early in the season, and mind you, we're also getting reports of influenza in the South Hemisphere, which is the beginning of the summer, which is not a good sign, because let me remind you that in 2009, the first cases of H1N1, which struck first in the South Hemisphere, were diagnosed in the North Hemisphere during the summer. And that was bad. So I don't want to scare everyone, but I can tell you right away that my dose of my influenza vaccine is already in the fridge at home, because I've been traveling for a few days, and I'm going to be immunizing my wife, my 2 daughters, and myself this weekend.
Okay. And by the way, I've done it already. I've had 4 shots, and I took also a flu jab, and that was within the space of something like 3 days before I traveled. And luckily, in my case, no side effect. Rachel, over to you. Rachel, you.
Thank you. Yeah. Mm-hmm. Thank you, everybody, and Pierre for the presentation. Very interesting. I had 2 questions. One was very close to the one that was stated earlier. So I'll turn it into maybe a request, if I may. I happened yesterday, as an anecdote, to talk to someone who refused to be vaccinated for COVID because she was afraid of all the side effects, etc. And interestingly, her punchline was to say that she read on the newspaper one article, and she knew someone, second case, who both died from a heart attack shortly after having got the vaccine. So long story short, I was thinking, if ever there is some data on side effects, and indeed there must be some, but certainly not as great as the positive effects of the vaccines, that could be something useful and that we could relay as well, because I think there's a lot of biases and opinions when it comes to those cases, and probably not enough scientific data that are spread across, let's say, normal people. And that if we can help.
Yeah, and you're raising a good point, which is, you find the roots of vaccine hesitancy are to be found in hyping stories. Some of them may be completely untrue, and ignoring the I would say educational communication from authorities, which tends to be a bit dull. And typically what is crispy and actually sells better than anything which is, fact based. I'm not even counting. I don't think this has happened recently. There's not one day when I'm not insulted on Twitter for being pro-vaccine. Okay, and you have people saying, I know several people who died from the vaccine. Say, wow, you know what? Ask for a pharmacovigilance survey, or please show me the results of the pharmacovigilance investigation. And then, of course, the discussion turns immediately acrimonious, and then they move to insults, and they don't have anything to present for their case. So. I think this pandemic has exacerbated the anti-vaccine sentiment, also because of the curtailing of the civil liberties that was put in place in the early days, where people were, locked down and had no access to certain activities, to restaurants, to etc. It was perceived as an intolerable encroachment on their civil liberties. Now, ask the Chinese. As the people in Shanghai who've been locked down without being able to leave their buildings, where the police would actually put chains and locks on the outside doors, so people were actually locked in the building. Can you imagine this, flying in Europe? Never. So I think there's a big question of perception, and there's a big question of, being able to look at the facts. It certainly takes a brave heart to take on a discussion with an anti-vaxxer. Now I have learned my lessons. I have to do it more delicately, because I tend to run out of patience a bit fast with these people.
Well,
Sorry, this is Seema here. Just a little on this. It was a great point raised by Rachel that what are the side effects? And I'm a pro-vaccine, but are the doctors and the healthcare professionals having a route to reporting the side effects? Because I haven't found one in my interactions with the hospital and the physicians. When they have a case where a person says this could have been a side effect, one of the doctors or a team of doctors got back to me saying it's not very clear. So if you don't report the side effects, how do you?
That's a very good point. And there's always, everybody knows there's underreporting. Typically, when a new drug is launched, and not just vaccines, at the beginning, say first couple of years of commercialization, you've got an inflow of PV cases, especially when things are rare and unusual. But then when you're, for instance, nobody's reporting anymore fever, swelling, fatigue after a vaccine, because it's very common and it does not actually bring anything. Myocarditis with the RNA, or the clotting effect with the chimpanzee adenovirus strain of the AstraZeneca vaccine were reported very well. Why? Because it was new and it was quite visible and it was very unusual.
Sorry, Seema, I think you muted yourself, but I think the question went through. Christian, you've had your hand up for a while, and also after that, also Shashika. Yep.
Yes, and in the meantime, I already also found the parking space. Nice to meet you, and thanks a lot, Pierre, for this amazing presentation. This is very, very helpful for me, because I'm a newbie in the I work in the pharmaceutical sector for around 3 to 4 years. I'm a lawyer by profession and working in marketing and sales. So this was very, very helpful, and thanks a lot for that. I'm interested in the R&D of new procedures and new drugs, and you mentioned as well that there are new therapies or companies with new therapies knocking at the door of the regulators as well. Funny enough is that a very close friend of mine, he's running a company called RocketVax Limited in Basel.
Yep.
They are partners with the Swiss TPH as well, with the university. And you talked about the storability, combo vaccines, and they are. So first question is, are you aware of them? Do you know them, Pierre?
No.
Okay.
But I'm going to be doing a little bit of research to check them out.
If you want, I can tell you some. I can give you some information about them. I got the deck from the CEO explaining what they do. So, longer lasting immune response resulting from the T cell activation. It's a nasal administration, and very interesting is that effective despite the mutation of the coronavirus spike protein, because it contains all viral proteins and not only the spike one. So I would be very much interested in your opinion on that.
Yeah, sure.
Salman, if you send around the presentation,
yep,
and I get the address, the email address of Pierre's, yep, I will deliver it to them. And sure, to the others, if you're interested, I can surely share it in a broader group.
Perfect. So just that you know, Christian is a member of our advisory board, by the way. He says he does not know much about life sciences, but that's not quite true. He knows a lot, actually, and he's bringing a different perspective to our group. So thank you so much for that. Yes, Christian, I will send with later on also the presentation, but also Pierre's coordinates. Shashka, you've been waiting patiently. Sorry,
yes,
and by the way,
so
I'm a little bit also conscious of Pierre, of your time, but I hope that you have a few more minutes.
Yeah. I have a hard stop on the half hour, but otherwise, until half past, I'm available.
Perfect. Thank you so much. Shashka, over to you.
Yeah. Can you hear me?
Perfect. Yes, we can.
Okay. Thanks, Pierre, for the great presentation. I have one question. So when you were talking about the mix and match importance to elicit it more intense in response, I was wondering if a person is vaccinated with a RNA vaccine, let's say, but then also have a natural infection, I mean, wouldn't that be type of a mix and match? And then the second question, the follow-up of that is, in this situation, what type of a booster is recommended, and does it matter?
Good question.
Okay, those are excellent questions, by the way. The short answer to your first question is yes. Typically, when and this is what I was referring to as immune imprinting, because essentially today it's impossible to find on the population someone who's COVID naive. Okay, either you've been exposed to the vaccine, or you've been exposed to the virus, or both. So, when you're vaccinated and then you get an exposure to the virus, you get what is called a natural booster. So essentially, your immune system is, put into a higher drive by the fact that it recognizes a pathogen. They say, well, that thing is, I've seen a cousin before and it doesn't belong here, so it's okay to act and react to it, and that is called a natural booster. Does this mean that you can waive the need for a vaccine booster? Probably not, because it is known that the coronavirus infections in general do not provide a very lasting immune response. So you do get a response, but that response is not necessarily lasting more than a year, if you will. So, and again, there's a lot of work ongoing, because you need to look at various subgroups, and depending on their history, what is the level of protection? And the only proxy that is used to gauge the level of protection is what is the level of immunoglobulins type G that you have, and how neutralizing are those antibodies to the currently circulating strain. So you do what is called neutralization assays, where you essentially take the plasma of the person and you check what is the amount of neutralization of the viral strain. That's actually does not measure what is the level of defense that you have on your mucosa at the point of entry of the virus, meaning IgA, and it does not, unless you do a complex ELISpot testing, which is not done in routine, other than, it's done in clinical trials for the what is called the immunogenicity cohort, but it's not done in routine in the population. Doing an ELISpot, meaning checking what is the library of epitopes recorded by your T cells. Okay, so to the second part of your question, is it still relevant to get a booster when you had an infection after the vaccine? The answer is yes. You can probably space the booster out a little bit more, meaning put it 6 months after the infection rather than 6 months after the previous vaccine shot.
Yeah, it was more what type of a booster would be recommended, and does it matter?
I mean, I would be provocative if I was speaking to an authority and say that it matters, because they tend to take a different approach. They say boost the population irrespective of the booster that we have, because this is how the policy is being built. I think the reality is the booster matters depending on your own history. And again, take case in point, take the situation in Israel. Israel, in exchange of being the first country, or one of the first country to receive the Pfizer vaccine, agreed to providing population-based data, okay, and reporting what was happening on the population, both in terms of epidemiology, in terms of serology. When you look at the latest papers, and there's a big cohort of several thousand subjects who received a 4th dose of the Pfizer vaccine, and they received only RNA throughout. 2 doses of priming, 3rd dose meaning first booster, 4th dose meaning second booster. 6 months after the 4th dose, half of them were not protected, were below the protective level in antibodies. Why is that? And this is published data, okay? Why is that? Because RNA is not stimulating the T cells sufficiently. And therefore, you have a response clearly, but it is not a lasting response. So I would argue that someone who's not had any viral vector would benefit from a better stimulation of the T cells.
Very interesting. Okay. Yeah.
Clear. Thank you.
Okay. Thank you so much, Reshka, for that question. Now, I think we're coming towards the end. And Pierre, you have left us, first of all, thank you so much for an excellent presentation, but you've also left us with a lot more food for thought. One of them, of course, one of them.
And you know what? And as soon as the presentation is there, I remain available to anybody in this group to answer, questions or for a dialogue. I mean, this is an ever evolving. I won't pretend I know everything, of course, because it's an ever moving field. But I tend to read a lot on this whole thing. Of course, I have my own opinion. And I'm happy to get into a discussion with anybody who's interested in doing so.
Thank you so much. And I can tell from personal experience that Pierre is very accessible, very open for discussion. I think, Pierre, that it was said also somewhere halfway through that we probably will have to have another session with you. This was the second one. We'd like to have probably sort of a few months down the road, have another session and hear more, from your words of wisdom. For me, it was interesting to look at a couple of things. Despite really the irrefutable evidence of the importance of vaccination, there are still some educated people who are still not convinced. And that is for me even more worrying. I mean, you said before that there's a gap between educated and uneducated, but some so-called highly educated people are still denying the usefulness of vaccination. I think going forward, that is something for we as a society that we should be thinking about, which I think is quite worrying.
It is, and actually, I'm a member of a couple of working groups on how to deal with vaccine hesitancy. In the specific case of COVID, I think you should start by saying, when someone is saying, "Oh, the COVID vaccine is not working," or "I don't trust the COVID vaccine," hang on. There are 4 different technologies. So what is it that you don't trust? You don't trust RNA because it was developed fast? You got other options, okay, with time-tested technologies that have, of course, their strengths and their weaknesses. So I think to me, addressing the anti-vaccine sentiment starts by bringing the conversation back into reality. Trying to break down the argument into smaller pieces to try to understand what are the real root causes of that person's hesitation. And because the reality is you have the real diehard anti-vaxxers are not very numerous, but you do have many people who are vacillators for good reasons. I mean, let's face it, a vaccine is a medical intervention, most likely a painful one, on a healthy person. So you better make sure that you're convincing about the efficacy and safety ratio.
Yeah. And I think, what seems to be, conveniently forgotten is that it's now almost 2 years or more than 2 years since we started getting vaccination. So there is actually a fair amount of user data, people who have been successfully vaccinated. So you cannot say in the beginning, of course, I think there was somewhat justified. I decided to ignore it, the fact that there's not enough experience. But now, today, we do have enough experience. Yeah,
I agree.
Yeah. So, well, with that thought, and I think, Pierre's message is very clear. Go and get your booster, please, and go and get your flu vaccine if you haven't done that.
Yeah, and in general terms, stay away from people who cough and sneeze around.
Well, I just came back yesterday from Frankfurt, from CPHI, and I was wondering, my God, was that a very wise thing to do? Because there were people coughing in the hall.
Exactly.
There were people coughing in the train on the way back. I'll be flying on Saturday to.
And wash your hands.
Exactly. And aeroplanes, by the way, this is something which is forgotten. Aeroplanes are generally quite safe because the HEPA filters which are built in.
Yes.
So if you're taking sufficient precautions, like, what's the big deal? I mean, put on your mask. But I think you should be relatively safe. Yeah.
Yeah, I would agree with that. Absolutely.
So on that note, once again, thank you so much. For an excellent presentation as always, Pierre, and thank you so much to all the participants. We'll make sure that we get Pierre's presentations and his coordinates over to you. All the best then.
Thank you very much, Salman.
Thank you.
Thank you all. It was a pleasure seeing you all.
Bye.
And stay safe.
Bye.
Thanks a lot. You too. Bye-bye.
Bye-bye.